Hi students, this is A. Mandari Kumarrajis. In this video, we are going to discuss MCQs on genetics, mainly based on Neet entrance examination. So please make use of this video and listen carefully. Let's discuss the topic. Which radiation induce the formation of thymine dimers in DNA that interfere with its replication? The option A, UV radiation, option B, X-rays, option C, gamma rays, option D, infrared light. What is called thymine dimers? Actually, it is a photolesins. Lesins means blisters. Blisters are nothing but நம்ம உடம்புல சின்ன சின்னதா வரும்ல பருமல். They are called as blisters. So thymine dimers are nothing but a photolesin produced by UV radiation. Produced by UV radiation. So the option is A. Option A, UV radiation is the right answer. UV radiation can form covalent linkage between consecutive bases, between consecutive nitrogen bases along the nucleotides. So this ultraviolet radiation can form covalent linkages between adjacent nitrogenous bases. So the option A is the right answer. UV radiation can cause photolesins, that is thymine dimers. So it can interfere DNA replication. The second question, which of the following radiation do not result in any mutational change? Option A, X-rays, option B, gamma rays, option C, UV rays, option D, infrared rays. So option A, X-ray can cause mutation. Option B, gamma rays can cause mutation. UV ray can also cause mutation. So the option D is the right answer. Infrared ray cannot cause mutation because it possesses low energy. Infrared ray contains very low energy. It is not able to break bonds of DNA in a cell. It is not able to break bonds of DNA in a cell. So option D is the right answer. Mutation can be induced by X-rays. Mutation can be induced by X-rays was revealed by dash. Option A, Stanley Miller, option B, Bridges, option C, Muller, option D, Darlington. So option A, Stanley Miller. Stanley Miller performed an experiment that may explain what occurred in the primitive earth a billion years ago. He sent an electrical charge through a flask which contains chemicals like methane, hydrogen, water and ammonia. This electrical charge reacts with these chemicals and it produces organic compounds like amino acids. So Stanley Miller explained how or what occurred in the primitive earth by an experiment. So option A is the wrong answer. Option B, Bridges. C.B. Bridges, that is Kelvin Blackman Bridges, discovered the crossing over is completely absent in some species of male Drosophila. So option B is the wrong answer. Option D, Darlington. Darlington, who discovered mechanics of crossing over and its inheritance. Darlington, who discovered mechanics of crossing over and its inheritance. So option D also wrong answer. So the remaining option is C, So option C is the right answer. Mutation can be induced by X-rays was revealed by the scientist Muller. Then the fourth question, which chemical mutagen results in deamination of bases? Deamination means the removal of amino group. Removal of amino group. So chemicals like nitrous acid, hydroxylamine, sodium acid can modify bases by deamination process. Can modify the bases with the help of a process called deamination. Deamination means the removal of amino group from amino acid and it converted into ammonia. So this nitrous acid is the right answer. This nitrous acid can modify the bases like adenine, guanine, cytosine. The adenine it can substitute adenine to hypoxanthine, then guanine to xanthine, then cytosine to uracil. This nitrous acid can substituting the adenine to hypoxanthine, then guanine to xanthine, hypoxanthine, then guanine to xanthine, then cytosine to uracil. Cytosine to uracil. So option A is the right answer. In a dihybrid test cross, the genotypic ratio of the progeny is, it is a direct question. So option B is the right answer. Option B is the right answer. In a dihybrid test cross, the genotypic ratio of the progeny is 1:1:1:1. The sixth question, find the correct match. Column 1, column 2. Option A, 2n minus 1 minus 1. B, 2n plus 1. C, 2n plus 1 plus 1. Then option D, not option D, 2n minus 2. In column 2, they have given that condition name. Trisomy, monosomy, nullisomy, double trisomy, double monosomy. So A tell us it is a double monosomy. 2n minus 1 minus 1. So A can match with the Roman letter 5. So option that A can match with the double monosomy. 2n plus 1. So this condition tell us the term trisomy. 2n plus 1. So that B can match with trisomy. 2n plus 1 plus 1. So this condition tell us double trisomy. So option C can match with the Roman letter 4. 2n minus 2. So this condition can be called as nullisomy. This condition can be called as nullisomy. So option D can match with Roman letter 3. So option 1 is the right answer. Option 1 is the right answer. With increasing age, the linkage gas, it becomes strong, becomes weak, terminates, remains unchanged. So linkage means a gene which is present on the same homologous chromosome. Genes are present on the same homologous chromosome and that condition is called as linkage. So that linked genes are remain together for more than 2 generations. The linkage is said to be complete linkage or perfect linkage, and that linkage, that bonding becomes so strong. So with increasing age, the linkage becomes so strong. Linkage becomes so strong. Then some factors like temperature. If the temperature increases, linkage decreases. The temperature can decrease the linkage between genes. Then X-ray. X-ray can also decrease the linkage between two genes. X-rays can also decreases linkage between two genes. 8th question. If there were only parental combination in F2 of a dihybrid cross, then Mendel might have discovered dash. So here they have mentioned the term parental combination. If the gene is linked, if a two gene get linked, we are able to get parental combination mode. So here they have mentioned the term parental combination. If there were only parental combinations in F2 of a dihybrid cross, then Mendel might have discovered a process called linkage. A process called linkage. So option C is the right answer. The 9th question. Mark the correct statement with respect to Mendelian dihybrid cross where two parents differ in two pairs of contrasting traits, that is seed color and seed shape. Option A, 3/4 of F2 plants have green seeds and 1/4 have yellow seeds. The phenotypic ratio of dihybrid cross is 9:3:3:1. Here they have mentioned 3/4 F2 plants have green seeds. Green seeds represents the recessive character. 1/4 have yellow seeds. So it represents the dominant character. So option A is the wrong answer because 3/4 represents the recessive character. So that is the wrong answer. Then option B. Mendel cross hybridized F1 plants and got 9:3:3:1 phenotypic ratio. Mendel cross hybridized F1 plants and got 9:3:3:1. So in F2 generation only, we are able to get phenotypic ratio 9:3:3:1. So option B is also a wrong answer. Then option D. Parental phenotype ratio is 1:1 in F2 generation. So it is also a wrong answer. So option C is the right answer. In F2 generation, round and ringle seeds shape segregates and just like in a monohybrid cross is the right answer. So option C is the right answer. Then the 10th question. Starch grain size in garden pea, flower color in 4 o'clock plant, and heterozygous individual for sickle cell anemia are examples of. Option A, incomplete dominance. Option B, non-allelic interaction. Option C, codominance. Option D, intragenic or intralocus gene interaction. So option D is the right answer. Option D is the right answer. The 11th question. Morgan carried out several dihybrid cross in fruit fly and found that dash. Option A, loosely linked genes show low recombination. Option B, the strength of the linkage between genes of white eye and miniature wing is lower than genes of yellow body and white eye. Option C, tightly linked genes show equal amount of parental and recombinant types in F2 generation. Option D, all genes segregate independently of each other and the F2 ratio deviates very significantly from 9:3:3:1 ratio. Actually, Morgan carried out several dihybrid cross in Drosophila melanogaster. Based on his breeding experiment in fruit fly, he proposed a theory called chromosomal theory of linkage. The theory states that the linked genes are present on the same homologous chromosome in a linear sequence and it produces more parental combination in F2 generation. In the cases like crossing over, the parental combinations are low. So option B is the right answer. strength of linkage between genes of white eye and miniature wing is lower than the genes of yellow body and white eye. So option B is the right answer. Then 12th question. The two eukaryotic organelles responsible for cytoplasmic inheritance are option A lysosome and mitochondria, option B chloroplast and lysosomes, option C mitochondria and chloroplast, option D mitochondria and Golgi complex. So option C is the right answer. Mitochondria and chloroplast is the right answer. The 13th question. Consider the following four statements A, B, C, D and select the right options for incorrect statement. Statement A. Mendelian experiment had large sampling size which gave great credibility to the data that he collected. So it is the correct statement. So option A is not a right answer. The statement B. Recessive allele influences the appearance of the phenotype even in the presence of alternative allele. So option, that is the statement B, is wrong, because that statements tell us recessive allele influences the appearance of phenotype. So dominant character will be expressed. But here they have tell us recessive alleles can appear in the phenotype in the presence of alternative allele, in the presence of dominant allele. So option B is the wrong statement. So option B is the right answer. Then multiple alleles can be found only when population studies are made. Option C is the right statement. Option D. In F2 generation of Mendelian monohybrid cross, the tall and dwarf traits were identical to their parental type and shows blending. Option D is also an incorrect statement. So here the options are, they have given option A and C, option B, C and D, option C, B and D, option D, B and C. So here B and D statements are incorrect. Option C is the right answer. Option C is the right answer. B and D represents the wrong statement. Then the number of linkage groups in a cell having 10 pairs of chromosomes are dash. Option A 5, option B 10, option C 15, option D 20. So linkage group can be defined as the total number of linkage group in an organism is equal to the number of autosomes in a haploid cell. Number of autosomes in a haploid cells. Here they have mentioned 10 pairs in a cell. 10 pairs in a cell. In a haploid condition, the cell contains 10 autosomes. In a haploid condition, a cell contains 10 autosomes. So option B is the right answer. 15th question. Mendelian dihybrid and dihybrid with linkage are respectively related with how many chromosomes? In Mendelian dihybrid cross, we are considered two different characters. For example, seed shape. If the seed is round, it is represented by the allele capital R, because round is the dominant character, so it is represented by the allele capital R. If it is wrinkled, the seed shape is wrinkled, it is represented by the allele small r, because it is a recessive character. So it is a one pair of alleles. Again, the next character, seed color. If the seed color is yellow, it is represented by the allele capital Y because it is a dominant character. If it is green, it is represented by the allele small y because it is a recessive character. So again it is a one pair of allele. Total two pairs. Total two pairs. While in the case of dihybrid with linkage, the genes are linked together. The alleles are linked together, closely linked together. So these alleles can be considered as one pair. This alleles considered as one pair. The option B is the right answer. So option B is the right answer. Two pair and one pair is the right answer. The 16th question. A dihybrid plant with incomplete linkage on test cross may produce how many types of plants? Option A 2, option B 4, option C 8, option D 1. So option B is the right answer. It can produce 4 plants, 2 recombinant plants, 2 parental combination plants. So option B is the right answer. Crossover value of gene A and B is 5%, while crossing over of G and B is 15%. The possible sequence of 3 genes on chromosome is option A, A, B, C, option B, C, A, B, option C, B, C, A, option D, both A and B. The crossing over between A and B is 5. A and B is 5. So here A will come, then here B come. Then the crossing over between B and C is 15%. The recombination is more. So here C will come. So option C, A and B. And another possibility is A, B and C. A and B, C. So option D is the right answer. Both A and B. Both A and B. The condition in which only one allele of a pair is present is known as hemizygous condition. Hemizygous condition. 19th question. Which of the following condition represents a case of codominant genes? A gene expresses itself suppressing the phenotypic effect of its allele. That genes are called as epistatic gene. Genes, those are similar in phenotypic effect when present separately, but when together interact to produce a different trait, that genes are called as complementary genes. Alleles, both of which interact to produce a trait which may resemble either of the parental type, that genes are called as incomplete dominant genes. Alleles, each of which produces an independent effect in heterozygous condition, and that genes are called as codominant genes. And that genes are called as codominant genes. So option G is the right answer. The 20th question. A gene that shows its effect on more than one character is called as pleiotrophic gene. Is called as pleiotrophic gene. So with this information, I conclude this video. If you have any doubt, please let me know. Thank you very much for watching this video. Thank you.